Good evening, everybody. I'd like to acknowledge the Gadigal people of the Eora Nation, the traditional custodians of this land, and pay my respects to the elders, both past and present. And we also acknowledge the Sisters of Charity, Mary Aitken Head Ministries. Before I start, I would just like to mention that we will be filming and recording this evening's proceedings so that we can share it with other members of the PKD community in other parts of Australia who don't have the opportunity to come to this wonderful facility. I'd also like to thank Genome One for hosting the evening. My name is Robert Gardos. I'm the chair of PKD Australia, and I'm really thrilled that we're able to have this evening where we're going to learn about something which is really cutting edge. It's changed the world, even in the short time that PKD Australia has been in existence, which is less than 4 years. While I think of it, there is a questionnaire which we would like you all to fill out. It asks you for your feelings about this evening, and your perceptions of this evening, both at the beginning when you arrived and at the end once we've had the presentation. So if you could fill it out, it would really help us to know how to do these sort of functions in future and how to improve on them. Now, a little bit of background as to why I'm interested in PKD. We don't have it in my family, but we were introduced to it, unfortunately, some years ago when a family where the eldest child was in the same primary school class as our son and played soccer together with our son. We learnt that the family had PKD. Every one of the 4 children had AD-PKD. And normally, every child has a 1 in 2 chance of having that. So when my wife and I learned about this, we thought to ourselves, we really have to do something, if we can, to find a cure to this disease so that these 4 children and anybody else like them in the world may benefit from it. So what we did was we went to Westmead Hospital, where we spoke to a couple of the senior people in the renal department, and we asked them what we can do. And the suggestion from Dr. Gopi Rangan, who is now the chair of our scientific advisory board, was that it would be possible to maybe get a PhD student to do some research into PKD. So we decided to fund that, and it was going really well, and we had updates on it, but it was going glacially slowly, and we felt that there wasn't going to be an answer in time for these 4 young people. So together with the mother of these young people, we thought about what we can do, and we decided to set up a charitable foundation. And we brought another family who also happened to have a child at the same local school, although that child was younger than both our son and the son from the other family. But this family had a different form of PKD, ARPKD, the recessive form. That young man was diagnosed in utero with the disease. So it's a disease that occurs much more rarely, but is very serious. And it needs also to have a cure found for it. So that happened, as I said, almost 4 years ago. We've been going in that time. We looked around at the time of establishment, and there was an inevitability about PKD. We were told not to offer hope to our community as we were forming it, and we just couldn't accept that. And now when I look around and I see what we saw now on our laboratory tour here, there is hope. There is huge hope. Genetics offers hope. The first drug for PKD therapy, admittedly only for AD-PKD and not for everybody, is approved in Australia. It's already available in other parts of the world, and hopefully very soon it will be funded in Australia. So there is hope. There's a lot happening. There's an amazing amount of research currently, which we are very proud to have funded some of it. In fact, one of the speakers tonight is the recipient of one of our grants for 2017, and she'll be talking about what has been enabled by her findings. So it's very exciting. I think there is going to be a lot of very good news coming. There's certainly, as I said, a lot of research being done here and elsewhere in the world. And one of the most exciting things happened only at the beginning of last month, and that was the announcement of one of the recipients of a 2-year research scholarship worth $160,000 that was won by an Australian research team led by the man that this room is named after, Dr. John Shine. And he is a superstar of genetics worldwide, but he's particularly a superstar in Australia. He was the president of the Academy of Sciences. So we are really thrilled that we were able to contribute half of that grant, and our US counterparts are contributing the other half of it. So that puts Australian research well and truly up there with international research, because he was up against grant applications from all over the world, and only if it had passed the US tests and reviews were they going to fund it, and only if an Australian researcher was agreed to by the Americans were we going to fund it. So we are really thrilled that we are funding it, and we're very excited that Dr. John Shine and his team are going to be doing research into PKD over the next 2 years. So that's a little bit of background to what we do. Unfortunately, due to the fact that the sound system isn't working, we're unable to show you a video that we have which talks about one family's experience with ADPKD. But I urge you to have a look at our website. It's on there, and it's pretty moving, but it's only one story, and there are lots of stories. Everybody in the room that has a connection with PKD has their own story, and it's very valid, and we'd like to hear from you. We'd like you to share it with us and share it with everybody else who has it, because you're not alone. So now I'd like to hand over to Nicole Schonrock of Genome One, who will introduce the rest of the speakers, and I look forward to hearing them. Nicole?
So thank you, Robert, and thank you very much to PKD Australia and to Garvan for organising this evening. And thank you all to you for your interest in knowing a little bit more about genetic testing. So as Robert said, this field is moving incredibly fast, and tonight we have 3 very, very good speakers and leaders in their field who I'm hoping are going to be able to answer a lot of the questions that you may have and give you a lot of insight. So we have 3 speakers tonight. Our first speaker is Dr. Amali Malawarachichi, and she is a very, very qualified superwoman who is actually not only a trained nephrologist, but she has also completed her training in clinical geneticist and now works at RPA. And she's also doing her PhD at the Garvan Institute. So her work has been supported partly by the PKD Foundation, and she's going to be telling us about how she is using whole genome sequencing in the diagnosis of ADPKD. So Marley, we look forward to hearing more about your work.
Thanks, Nicole. Thanks everyone for having me. It's great to be here, and hopefully we'll be able to educate you a bit and hopefully keep you interested as well. So I'm going to speak a little bit about genetics in the kidney, and specifically about polycystic kidney disease. And I thought I would start by talking, asking the question, why genetics? Why suddenly all the fuss about genetics, and why are we talking about this? And what does it mean to you as patients and families who are affected with PKD? And I'll talk specifically about the genetics of polycystic kidney disease and some of the work that we've been doing here at Garvan about in developing a test for polycystic kidney disease here in Australia. So why genetics? We know that cystic kidney diseases often have a really strong inherited component to them. And there's a number of reasons why understanding inheritance can be helpful. To understand the cause of disease, to help answer that question, why did this happen in me? To understand how a condition is passed on. Those questions like, are there other people in my family that are at risk? Can I have a relative be a kidney donor for me? Or will I develop the same disease as my family member? And of course, genetics can help us to understand a bit about prognosis, about what's likely to happen in the future for someone with a particular condition. Before I talk a bit more about genetics and some of the research that we've been doing, I thought I would have a little overview, a little step back into high school biology, just to make sure that we're all on the same page with these genes and chromosomes. So you guys probably remember from school biology that all of the cells inside our body have a nucleus inside them. And when we look inside that nucleus, it's made of chromosomes. And these are what our chromosomes look like if we have a look at them under the microscope. And they're in these little pairs, and all these little sticks are chromosomes. And when we have a look at our chromosomes in even more detail, they're made up, if we unravel them, into tiny little components that we call genes. And our genes are very specific instructions for one very particular function in the body. So I kind of think of it like our chromosomes are this recipe book, and the genes are all of the recipes inside that book. And when we have a look at those genes or recipes in more detail, they're actually a sequence of letters that our body very cleverly decodes from that set of instructions into something that can make the proteins and all of the different structures within our cell. And if we get a difference or a mistake in one of these genes, they can result in particular inherited conditions. For polycystic kidney disease in particular, like Robert says, there's kind of two main conditions that we often think about. One of them is autosomal dominant polycystic kidney disease, ADPKD, and that's usually caused by differences or mistakes in one of two genes called PKD1 and PKD2. And then there's ARPKD, autosomal recessive polycystic kidney disease, and that's usually caused by a difference in a gene called PKHD1. I don't need to tell all of you guys that all of our families are different, and so inheritance patterns in families are all different as well. And you can see this is a few different families. One where there's lots of people who are coloured in that are affected with a particular condition in lots of generations of a family. And there's another family here where there's just one person who, out of the blue, with no one else having a family history of a condition, are affected. Or this other family where there's two children that are affected, again, with no one else in the family. And so we know that how we inherit disease in families varies. And some families inherit conditions in what we call a dominant manner. And we have two copies of most of the genes in our body, and sometimes a mistake or a difference in just one copy of those genes is enough to cause a condition. And this is what happens in ADPKD. And in this family, there's a person who's affected and their partner is unaffected. And when they go to have their children, they have a 50% chance of having a child who's affected with the condition, who inherits the gene with that same difference. So that's what we call dominant inheritance. But some families inherit conditions in what we call is a recessive manner, where you actually need a difference in both copies of a particular gene to have the gene have the condition. And this is what happens in ARPKD. And in this family, we have two parents who are healthy carriers. They have a difference in one copy of the gene, but another copy of the gene that is working as we would expect. They're well, but when they go to have their children, they have a 1 in 4 chance of having a child who inherits both copies of that gene difference and therefore develop the condition. And sometimes things can happen out of the blue, that we can develop a genetic condition when no one else in our family has one, and it's a change that occurs as a new thing in a person. And this is actually not uncommon in ADPKD, and about 10% of people with ADPKD are the first people in their family that are affected. And sometimes, despite our best efforts, we don't understand what's going on, and we can't identify a particular cause or a genetic cause for a condition. And that's where places like this have a lot of work to do to try and understand much more about a lot of these conditions. So given that we know quite a lot about genes and chromosomes, and there seems to be lots of good information to be had from our genetic information, why don't we go about and sequence everybody? One of the particular challenges in AD-PKD is that the sequencing is not very straightforward. The genes that cause AD-PKD are really big. There are lots of chapters in that recipe book, and so that's a challenge in itself to be able to sequence. But even more than that, the PKD1 gene has something called pseudogenes associated with it. And they're a pretty interesting little a component of evolution, where sometime back when we were primates, that particular PKD1 gene, that set of instructions, was copied several times in one of our chromosomes. And those copies are not functional. They don't, as far as we understand, do anything, but they really confuse our sequencing machines when we try to sequence the DNA. So when we sequence DNA, we take the whole string of DNA and we chop it up into little pieces, kind of like taking a puzzle and taking it all apart. But the problem with having those pseudogenes is that all the puzzle pieces end up looking exactly the same. And so it's a bit tricky to know where the puzzle goes back. And that has been a big challenge in diagnosing PKD genetically for a long time, and it used to cost a lot of money and require quite specialised techniques to sequence. So our research lab wanted to have a look at this because PKD is really common, and we were very keen to be able to develop a testing service that could be available for patients here in Australia. And the Garvan is really uniquely placed with these amazing whole genome sequencing machines to kind of address some of these problems. So we wanted to test whether whole genome sequencing, which is a slightly different type of sequencing, would be a better way to diagnose PKD. And you guys have been in the lab, so I don't need to show you this picture, but this is some of the sequencing machines that do some incredible work in sequencing the DNA very rapidly. And so what we did was that we took a group of patients who we knew had a clinical diagnosis of PKD, and we tried to see if we could identify what their genetic diagnosis was. And we found that in 90% of the patients, we were able to identify the cause using the whole genome sequencing technique, which we were pretty happy about. And we were very happy that this was published a few years ago in one of the genetics journals to show that this was a new technique to be able to diagnose PKD. But what we were really interested in is having a test that's available for people to use. And so after that, we thought, well, is this enough evidence for us to be able to offer a test for families? And we wanted to have a bit more evidence than just those initial 40 patients. So we partnered with the Mayo Clinic in the US, who probably have one of the largest research groups for polycystic kidney disease in the world. And they sent us about 40 samples of their most difficult to diagnose patients. They didn't tell us what the answer was, and they said, let's see if you guys can figure it out with your new technique. And we were able to identify the cause in 40 out of 42 of those patients. The other two patients, it's kind of a long story, but the testing was not really set up to identify the differences in them. And that was a study that was supported by the PKD Foundation Australia, who gave us a grant to be able to go out and do these really important validation tests that can make us more confident of the results that we give back to families. And after that, we thought, okay, we're ready for some diagnostic testing. And we've now had a diagnostic test available for PKD here through Genome 1 for about the last year. And we've tested nearly 100 patients now from across the country. And it's the first polycystic kidney disease test that's been available in Australia, and actually the first whole genome sequencing test for this disease in the whole world. So we're very proud to be able to offer it, and it's really exciting. So I'm going to finish now because there's lots of other people that want to talk. But I'll finish by saying that genetics is not all about the tests. And I think as doctors, we have a bad habit of loving tests and wanting to talk a lot about tests and investigations. But one of the things about genetics is that it's really personal, and that's, I guess, the crux of it all, and that all of our genetic stories and our stories of our diseases are all different, and that it's important that everything is tailored to each individual and their own story. And thinking about genetics is not just thinking about whether a test is appropriate for you or not, or whether you want to have a test or not, and there's lots of different answers to that. There's many other components to that. So I'd encourage you to think about talking to your local genetics service about those things so that you can understand what's relevant to you and make sure that you're getting your story read. So how do you find out more about the genetics or what's happening in your family, start by talking to your nephrologist and your GP. And there's some fantastic genetic services available all over New South Wales, which I'm not going to list, but there's a centre called New South Wales Centre for Genetics Education. If you Google it, there's some fantastic resources there of places that you can go to be seen to talk about genetics if that's something that you are interested in. And in New South Wales, there's a number of particular renal genetics clinics that are available as well, and Andrew's going to talk a little bit more about that next. So I'll finish there, and I think we're going to have a question and answer session at the end. So leave, I can answer questions now, otherwise.
Yeah. Is there any urgent questions for Amali?
It's not urgent. I'm just curious. Did you say you're the only, this is the only place we can have genetic testing done for PKD?
In Australia, yeah. Yeah.
Talk to you later about, do you work with Felicity Collins?
Yes, I work in the same department as her.
Okay, could I talk?
Yeah, of course. All right.
Thanks very much, Amali.
No trouble.
Great. So I have great pleasure in introducing Dr. Andrew Mallett, who's coming to us all the way from Brisbane tonight. So Andrew is a nephrologist by training with a big interest in renal genetics. He has been absolutely instrumental in translating genetics and genetic research into the clinic. So he's also the director of KidGen, the national director of KidGen, which is the renal flagship for the Australian Genomics Society. And as I said, he's really been driving making these tests accessible to patients. So Andrew, welcome and thank you very much.
Thanks, Nicole. I'm just Andrew. I'm from Queensland. That's a lot to live up to, I think. I'm wearing jeans because it's Jeans for Genes Day today, and I hope many of you have had the opportunity to do that today as well. So my remit today is to tell you a little bit more broadly about how we're integrating genomics and genetic health into kidney medicine around the country. And I think that's intrinsically related with polycystic kidney disease, because I think there's a lot of issues that are very similar, and I think the work that Amali's done, I think has been really trailblazing for us, because as an adult kidney specialist, effectively half of the patients that I see in my clinic that we operate have polycystic kidney disease. So I think, this is a really important thing to set out at the beginning. Well, why the kidneys? I'm a nephrologist. I live for kidneys. But for many other people, they're like, why? So what? I think the important thing about kidney health is that, 1 in 8 Australians actually have kidney disease, and I don't think that that's as clearly recognised as it could or should be. And actually, when you drill into, how many of those patients actually have inherited kidney disease or a genetic form of kidney disease, about 10% of adults and up to 50% of children with diagnosed kidney disease actually have an underlying inheritable or genetic form. There's actually emerging evidence that children who are on dialysis under the age of about 10, it's thought that near 100% of that population probably actually have an underlying genetic form. Unfortunately, kidney disease is also a very great equaliser. So when a patient commences dialysis, regardless of at which age, their likelihood of not being alive in 1 year is 100-fold increased compared to other people their age who just don't have kidney disease. And our best treatment that we have available today, kidney transplantation, which is an amazing miracle, does pretty well, but it only decreases that magnum of difference tenfold. So people with kidney transplants still have a tenfold increased risk of not being alive in 1 year compared to other people their age. And this is a story that has played out all around the world, and unfortunately has meant that I think kidney disease has been underappreciated as a major population health emergency. And within that, absolutely, inherited and genetic forms must be considered. So back in 2016, I apologise for so many words on the screen, but a big bunch of us, and kidney medicine is a bit unique in that actually it is a great specialty to be in, because you actually can get together a whole lot of people with a whole lot of different interests. And we got a bunch of us, adult and paediatric specialists, as well as genetic counsellors, clinical geneticists, our laboratory colleagues, both diagnostic laboratories such as here at Genome 1 and research laboratories, and said, well, this is a problem. We've got to do something about this. And we formed this thing, the KidGen Collaborative, which is the National Renal Genetics Initiative, and set ourselves a goal that within 3 years, from the middle of last year, so this is coming up middle of next year, we would transform the care that Australian patients would be able to receive. That the core of this would be multidisciplinary clinics where availability of genetic testing would be provided to those in whom it was indicated and wanted it to be done, and that we could do better and begin to transform change. We can't begin to treat the things that we don't understand, so this would seem a logical first step. Given that 3 years is the middle of next year, we've still got a little bit of work to do. But there actually have been some major successes. We've had a really fortunate time to partner with a lot of organisations. We're predominantly working with Australian Genomics, but partner with patient organisations such as PKD Australia, as well as Kidney Health Australia. And we've gone from literally starting with our one clinic in the Royal Brisbane in August of 2013 to, as of this month, I think we're at about 15 clinics around the country. So our clinic in the Northern Territory opens this month, a paediatric service in Adelaide opens also this month, and a service in Tasmania is opening in the next 2 months. So this has actually transformed the landscape in many ways in a very short period of time, and I don't think I'd ever really predicted that would occur. Here in New South Wales, obviously, there are 4 leading centres, and I think differently to other things that have perhaps changed in the healthcare environment before, these are teams who are all talking together and working together to change how we do what we do. So it's not different clinics working in isolation. We also, when we're trying to, organisationally work out how we would do this, it's important that there's, jurisdictions around, different states being involved, but also importantly, there are different considerations, not just research, but also in the clinical domain, registry efforts, collecting information, ethics, I think, and governance, I think is incredibly important. And actually, this has been a really great way for us to ensure that actually patients and families, like many of yourselves, remain the core of exactly what we're doing. From this has spawned lots of different projects doing lots of different things. And I won't delve into each and every one of these projects individually, but effectively we've found that actually by engaging with patients such as yourselves with potential inherited kidney disease, there's actually lots of unanswered questions. And we need to do something about that because we're interested in it. Many of our patients and families are interested in it. And I think together we're going to be able to make a big difference over the next few years. The one major thing I think that also ties us all together, I think, has been access to testing. And one thing that's astounded me is that in every state and territory thus far that we've begun to operate in around the country, the local rules and lay of the land in terms of access to testing is a little bit different. I think that's fairly fair to say, would you say, Marley? It's just a bit different everywhere. And one of the major things that's missing is there is not an MBS item number. So when you go to a doctor and they perform a procedure or order a test, often there's an item number. Because there aren't item numbers for genetic tests, it means that this becomes a little bit less certain. And that's why different doctors and clinicians might say different things to you about whether a genetic test might be done or not. In essence, though, today, for all of the patients that we see in these clinics around the country, if there's a clinical indication, testing can be provided. So please be reassured. But to our federal government, what isn't measured doesn't exist. So the first thing we've set about doing is actually in a flagship model, measuring what's coming through our large clinic network so we can quantify benefit, so that we can demonstrate to the government what the benefit is in the very naked aim of potentially going for what we call an MSAC application to potentially get MBS listing for genetic testing in the next few years. That, I think, will hopefully be transformative, and it's the first step, I think, down the pathway of integrating genomics and genetic testing into healthcare as it particularly pertains to our kidney patients. When we started this project, when was it? About 18 months ago. It's been a long 18 months, I think. I didn't actually think we would ever get to our target of 361 patients and recruits around the country. But as of today, we're actually at 70% of our target and actually on track to complete at the end of the year as planned. So if any of you have been in those clinics, thank you very much. Your participation and on the side of your clinical service will help us to generate evidence, hopefully to benefit both yourselves and others all around the country. I put this up there because it's complex, much like many of our patients. When we started out, we thought, oh, it'll be a cinch. We'll be able to fit all of our patients into nice, clean little boxes. We were very wrong. The patients that we see in clinical practice are wide and varied. And I think that from this, we've really had to come up with new and different ways to work out the kind of benefit the patients might experience and what kind of trajectories that they experience going through our health system in terms of whether a test is undertaken or not, whether the result comes back positive or not, or other. And I think that this has been really important for us, perhaps in our ability to engage with the community as well. Because before it was thought that this was all just very straightforward. There were patients, we saw them, that was the end of the story. But it actually is only the beginning. We have been able to slim down that workflow quite nicely. And in all of these clinics, they all operate a little bit differently. And that's a good thing to suit their local environments. But importantly, all of these clinics generally operate with an intake clinic or meeting, which is multidisciplinary in nature, where referrals are discussed prior to a patient being seen. You wouldn't go into a game of rugby without a game plan, nor should we go into a clinical consult, potentially with a complex clinical scenario with a similar kind of game plan of what information do we as clinicians have to have and have looked up beforehand. And then, obviously, within that clinic, a lot of issues might be deconstructed and discussed, genetic tests offered, and then the results come back, and we have to follow up on those results and communicate them effectively back to patients and families. But everything doesn't also go to plan. So what we found in some places is that the way these clinics operate is a bit different in different places. And that's actually a really good thing. Here in New South Wales, there have been some differences in different places, which is a good thing. Some services have been actively adopting genetic testing and genomic testing early, and so often these clinics are seeing patients in terms of returning results. The signal that we actually saw that began to alert us to this was actually that recruitment wasn't occurring or appeared to not be occurring in some sites. And so now we're actually going back and working through what are actually the experiences that are happening, why are those practices a little bit different, and why have they come to be. I think it's through understanding difference that we're able to actually inform what we should do next. And this has been, rather than an opportunity to go, oh, those buggers in New South Wales aren't recruiting, it's actually an opportunity for us all to improve our practice. I don't need to tell many of you that we have a complex health system. Would you believe this is the simplest representation of the Australian health system I have come across? The division of how it is funded, how it is operated between the different levels of government and the different organisations, public and private, is difficult for me to follow as a person who works within it, as opposed to patients. So we think we actually have to understand this a bit better and understand what are the things that make health service utilisation different for our patients with inherited kidney disease. And this is fairly new, so forgive me for going through this data. I'll skip to the bottom line. Effectively, in Queensland, we have a large registry of patients who have not yet commenced dialysis who have kidney disease, approaching about 9,000 patients from all across the state. We're able to have a look in that registry and actually identify patients who have known inherited or polycystic kidney disease and compare what health services they've accessed over the last 5 years in a de-identified way. And then Queensland, being a generous state, our health system has actually also donated an additional 27,000 health records of de-identified other patients for us to compare against. What we've identified is that patients with polycystic kidney disease who are at earlier stages of kidney disease actually utilise our health services more. So this is really a very hidden group who actually doing things about helping them is clearly indicated because our government really does. Our government and health services, they respond to expenses. What needs to be provided and paid for. And here we're beginning to nut out exactly the details of how that's occurring so that hopefully we can begin to reimagine our health services to target the real need that's underlying this. The next step that we're planning on doing is actually going and drilling into what are the reasons that these patients are coming to hospital, to try and identify any themes so that we can actually do something about it, okay, and help people before a hospital stage to help them remain independent, healthy, and in the community. Diagnostic testing, I think, has been a massive change, and when we first started out, and this was before the work that Amali has done here at Genome 1 and the Garvan, when we started at our first clinic, all of our genetic testing we were sending to the United Kingdom. We were great exporters. There was nothing onshore. And we didn't think that was exactly right, much as obviously was presented just before. So a service was commenced at Kids Westmead, not very far from here, which actually has operated as a very competitive service. And to give patients expectation of return, I think, has been critical. Effectively, that service, depending on the diagnosis, on average provides a genetic diagnosis at about 43% of the patients that we see. And that's important because actually you order genetic tests for different reasons in different people. Sometimes you're actually expecting a negative result. So a 43% diagnosis rate isn't a bad thing. In fact, actually, that's often a very good thing. When you start doing testing at scale, as we've been doing around the country in this clinic network, you start finding individuals and families where you haven't found a genetic cause. And so research genomics, I think, has been a critical element as well. To date, we've actually recruited about 50 families around the country, including some from here in Sydney. About a third of those families to date have actually had a genetic cause identified, which is often an ultra-rare or recently reported inherited kidney disease, which would not otherwise be able to be tested in a clinical setting. And this has been important for us because a really great example was one individual family who have a recessive form of kidney disease, where no matter what counselling was provided in the absence of a clear genetic result, the mum of this little boy thought there was something she had done. And it was only actually about 3 years after the result of the study participation had been returned that the mum actually came to clinic. And I remember when she said to me, you know, I finally understand that this wasn't something I did. And even despite all of the counselling that had been provided to prove that, no, this wasn't anybody's fault, it actually took some time for that to actually sink through. And I think it's when you see moments like that it makes all of this kind of work really worthwhile. This is an important family who we've seen who have a related condition to PKD, a tubulointerstitial and cystic kidney disease. And in fact, I met these two boys in my first year of training as a renal registrar. And the third question that they asked me is, will we pass this on to our children? Because they were in their late teens, early 20s. And as you can see, with all the coloured boxes, in that family tree, lots of people have been affected by this kidney disease, which causes early onset of commencement of dialysis. We tested for all the known causes for the kidney disease that was presumed that they had, couldn't find any answers. It turned out in 1982, which was a year before I was born, they'd actually been studied in depth and kidney biopsies undertaken, which demonstrated the underlying diagnosis. They'd actually forgotten they'd had kidney biopsies, which was bizarre, because that's not something you forget in a hurry. We collaborated with a group of colleagues in Cambridge in the UK and actually identified that they had a mitochondrial form of kidney disease. And we proved this in a few different interesting ways, but the important thing here was that we could give an answer back to those boys that actually know, because this is a mitochondrially inherited condition, their children aren't at risk. And so they've actually now gone on, and the eldest of those two has actually begun a family in the last few years after receiving a kidney transplant. There's some amazing work happening in Australia, I think, all around the place. So these two people here at the bottom are Amalia and I are really fortunate, I think, to collaborate and work with, Melissa Little and Ian Smythe, who are veritable rock stars in kidney research. Melissa has done the work to generate a protocol to make mini kidneys. And actually, you know, it's a big thing. Took out the cover of Nature magazine about 2 or 3 years ago with this work, which is just, it doesn't get any higher really than that. Ian works with mice, which isn't nature, but is still very important. And we're able to use these two model systems to model inherited kidney disease, to try and work out, well, how does a disease work? And also look at things like drug screening. And hopefully, in the 25 to 30-year time period, there may be bioengineering options as a result of this, so growing new kidneys or the like to help patients. Related to polycystic kidney disease are these recessive cystic type kidney diseases, one of which is a condition called nephronophthisis. Now, ask 5 nephrologists how to pronounce that. You'll get 5 different ways. I apologise. NPHP for short. A young, very brave lady from Queensland who's about 12 now participated and through our research whole genome sequencing, we're able to identify the underlying cause for her kidney disease and actually change the diagnosis. And then using a small sample of skin cells, fibroblasts, we're actually able to correct the genetic change that's caused the condition and make small mini kidneys, and actually study that in detail and prove that this is a valid way of being able to study inherited kidney disease. Here on the right, we've actually been able to study a different form of nephronophthisis in a young patient and family from here actually in Sydney, and actually get to the bottom of, well, what's the primary defect that's going on in the kidney cells to try and help direct what treatments might be potentially looked at in addition in the future. I remember being in year 12 in 2000 when this was the cover of Time magazine, when the first draft human genome was done. I think that was the first draft, Ben, and it was a couple of $1 billion in 10 years it took to get to that point. $3 billion. Oh my God. So nothing much, not much time. We've all got 10 years to wait. Not really. But what's happened since then has been phenomenal. So the decrement in unit cost, so the dramatically reducing price it costs to undertake whole genome sequencing in a laboratory setting, has really been transformative. And this has happened far faster than everything we've seen before. And the previous high watermark was the rate of change of computing power in computers. We all remember those days where you buy a computer, you've barely taken it out, and whoops, there's another one that's twice as good for the same price. This was actually outstripping that. Here in Australia, we have I often call it our dirty little secret, but amongst those who have end-stage kidney disease, and we see these sometimes in our clinics all around the country, about 5% of those patients actually, we have no idea why they're on dialysis. We have no idea what's caused their renal failure. And whole genome sequencing as a transformative technology provides us options to reconsider this, and potentially to find causes that may not have been previously appreciated. And so here in Australia at the moment, we're undertaking a large national study to try and do this called the HIDDEN Project. And actually, I think quite proudly, the very first case was only launched on the 1st of July, and the first case was proposed and accepted here in Sydney by Amali this past week. So we're going to be enrolling about 200 patients around the country, and we will be pushing whole genome sequencing to its very edge of what it can achieve, and hopefully we'll be able to help real patients. It's a complicated workflow within the clinics that we already have, but effectively we're going to be working with a lot of different partners to do everything you possibly can with this technology to try and find answers that may not have been previously appreciated. A shameless plug, because we have an annual symposium for renal genetics. That was the other thing. You know, a lot of people are doing a lot of work in this field, but there weren't a lot of dedicated meetings for people to get together and present their findings and engage with each other. So we commenced this meeting a few years ago, and this year it's actually going to be happening in Sydney with an absolutely amazing lineup of speakers, and it's very capably organised by Amali, Steve Alexander and Hugh McCarthy here in Sydney. I'm really looking forward to this program. It takes a lot of people and a lot of work, I think, to do this kind of program at a national level. And we've been really fortunate to have partnerships with organisations like PKD Australia, Kidney Health Australia, as well as international partners like Genomics England, that I think have really helped us to work out what the best way forward is, and also to begin helping us to resource how we can do this kind of footprint. So just to reiterate, you know, the national map of where we're at today and how we got there, the plan is that by the end of this year, all states and territories in the country will have a presence of a subspecialist or quaternary renal genetic service, apart from the ACT, but we won't mention them. Most of all actually will be here in Sydney and in Melbourne with 4 clinics apiece. So whether you're an adult, a parent, a child, or you're at risk of an inherited kidney disease, there is a service that your GP or kidney specialist can refer you to receive subspecialist advice and to support your own kidney specialist in your care and management and counselling. So I think that's been a really amazing achievement to get to in such a short period of time. Where do we get to from here? Now, I'm not a Trump kind of fan, but I just couldn't help myself. So as I said at the beginning, I think kidney medicine has been really underrated, and I think this is one of the great opportunities for us to do better in making kidneys great again. In the next 25 to 30 years, I have every confidence that actually the way that we replace kidney function is going to fundamentally change. dialysis will be the thing that I hope in my professional lifetime is the thing we used to do, not the thing that we continue to do. In the interim, though, we have to live in the reality we have, and I would dearly like for every patient who I look after who's on dialysis or nearing it to be able to get a kidney, but that isn't feasible today, and thus supporting things like the Donate Life program and new initiatives to like clinical trials to slow down kidney function, I think are clearly things that we should be backing to help our patients get the best benefit. I just love Oprah. Couldn't not put her in. And I think for me, this 3-year goal has been ambitious, and we've had a really great opportunity to partner with a lot of clinicians and collaborators around the country, but it really all does come down to each individual family that you see. The things that they tell you, that people like yourselves tell us, I think are always unpredictable, and that's great. I always like being surprised in clinic by people's experiences and the questions that they ask, and I hope you're thinking up some curly ones for the end of tonight. But it is really important to think that actually this isn't just about individual people, this is about families. And they're all around the country, and they're not picky about where they are, and we have to do better as, I think, a healthcare community to provide benefit for all of them. So with that, I'll close and let you think on some questions for the end.
So our final speaker for this evening is a very experienced genetic counsellor who's currently working at Genome 1. So Marianne Young is probably, I think, one of the first 4 genetic counsellors that ever existed in Australia. So she's practically invented this profession, and it is incredibly important. She provides that link between the patient and the doctors, and can really answer a lot of your questions that you have tonight. So I'm hoping that she's going to be here to answer some of those questions that you may have and give us a bit of an introduction.
Thanks, Nicole. She makes me sound incredibly old that I've been... It is a very new profession, I have to tell you. So we've heard tonight about sort of the research and PKD and how we've come through now with a genomic test. And Andrew's really nicely painted how in Australia this is gaining more traction, I suppose, and it's through genetics that some of that is happening. But it's also through families like yourselves that it's happening as well with the traction. So we can get a bit tantalised at times by the technology. It is very tantalising. I am not someone who's into technology, can I tell you? I am hopeless. But I think it's wild at times. I do tours with Ben and my head just gets blown off. But we must remember that it's not about the technology, as Andrew was saying. It's actually about families and what happens in families. And the reason I suppose there's been this rapid development of genomic testing is to help in making a diagnosis for people, because it's in making a diagnosis that then you can go forward in how best to treat people, drug therapies, lots of different things. And it's actually about improving patient and family care. At the end of the day, that's what this is all about. And it's actually about treatment, and hopefully one day it's about prevention. Nicole asked me a question that some of you might have about why do people have to go to these clinics, and why can't a GP order a genetic test for polycystic kidney disease? And it's actually a really good question. But primarily delivering information, people have gone to clinical genetics services, and now what we're seeing is the evolution of these what we call multidisciplinary clinics where people can go. So it's a bit like a one-stop shop. So it's for your benefit that these clinics have been, I suppose, they're evolving. So you can go along and you can see Andrew, and he can talk about the kidneys and also some of the genomics, and then you can see Amali and she'll talk about something else, and so you just have to come once and you get to see everybody that you want to see. So it's a great idea. And basically genetic testing, as Amali was saying, people come to clinics not just for genetic testing, they also come just to talk. And they come for what we call genetic counselling. So the first thing I just want to say is that genetic counselling is not therapy, okay? It's not sort of psychological therapy, okay? So don't be frightened of the term counselling. I've put this definition up here, but I just want to show you that it's actually a lot more than just offering genetic testing. So what happens when you come to a genetics clinic or you go to one of these kidney clinics is that people will ask you about your own history of kidney disease. They'll take your family history to see if they can work out, could this be a dominant form of polycystic kidney disease? Could this be a recessive form of polycystic kidney disease? Or could this be a new event in the family like Amali was talking about? So there's an assessment and an interpretation done. And then they talk to you about the way this could be inherited in your family, if it is an inherited type. So is it a dominant one or is it a recessive one? They talk about how best to manage the condition, what you can do about it, and also talk about genetic testing and whether that might be something that's helpful in your family. So genetic testing is actually not something that might be offered to all families because it may not be appropriate, but to many families it is. And the main aim of, I suppose, providing families with information is so that they can make informed choices about what they wish to do. Rather than, I suppose, the old system where the doctor used to tell you what to do, now we're trying to empower people to actually contribute to their decisions about the way forward in regards to their future health and regards to their family's future health. The reason a GP can't order a genetic test for polycystic kidney disease is it's actually really complicated. Genetic testing is complicated. It's not just like looking at one gene. I think as Amali showed before, the genes are big, they're complex, and we get complicated results. So sometimes when you test someone, you will see a change in the PKD gene that you know is the cause of the renal disease in that person, and that's easy. But often you'll find something that you don't actually know what it means. So it could be associated with the condition, but it may not be. And we don't have enough information at this time to tell us whether it is or not. And what we would do is keep looking for that information to see if one day we can give people more information about that change that's been seen in the gene. And sometimes we just know that what we're seeing is not associated with the disease. So it's actually not just a matter of yes or no. There's a lot of grey in between, and that's why we have experts like Thomas down here who can spend hours, I don't know how he does it, but looking at these genes on a screen to see what changes are happening and to determine whether the changes he's seeing are actually causing the PKD. The other reason I suppose GPs can't order a test, I'll go to the bottom one first, is it's really expensive. And our healthcare budget can't afford tests to be ordered on people who it may not be the best test for that person. So to do a whole genome sequence for someone where you think they've got polycystic kidney disease, Ben, $4,000. For this one, it's about $2,500. So that's quite a lot of money. So you wouldn't want GPs just to be ordering polycystic kidney disease tests for every second person that comes in, or our health budget would be absolutely shot, basically. The other complicated fact is that once you find a variant in someone that you know is causing the PKD, you can then offer genetic testing to other people in the family to tell them whether they have inherited that variant that's causing PKD or they have not. And so that requires discussions in the family. There's a lot of issues to talk about because if you're in a large family, some people might get it, some people might not. How would that be? Parents often want to talk about that, you know, they might be a bit nervous with their kids going through what we call predictive testing because they just get nervous because it's not nice to hear that some of your kids might have inherited this condition or the gene for this condition. And the other thing is around communication in families. It doesn't always go as well as you think it's going to go, and sometimes it's quite tricky to actually communicate genetic information in families. So that's really why a GP can't order a genetic test. And as Andrew was saying, most genetic tests can't be ordered by GPs. There's a few that can, but most have to be ordered by specialists like Amali and Andrew. Yeah. So that brings up the issue, I suppose, about equity and access in our healthcare system. And I know one of the other questions Nicole asked us to address was why in some states can people actually get this testing paid for and why in other states can they not? And unfortunately, we have this healthcare system that is disparate, and it depends on what state you're in as to actually what you can access and whether that is paid for or not paid for. It's not to say it's right, but unfortunately it's the way our healthcare system is delivered. I think also in my history of working in genetics that the PKD, in specialist clinics, we haven't seen a lot of families coming forward with PKD. But I think that's going to change now, and we're actually going to see more people coming through with PKD who will actually want genetic testing. And so the issue of equity and access is going to have to be addressed so that everybody can have a test for PKD if it's needed, and it will be paid for by the health system. So Amalie and Andrew have both talked about that the human experience actually influences genomics, and what that means is living in your family and growing up influences the way you think about polycystic kidney disease enormously, and every person you talk to will have a different experience. It's like just the stories you have of a kid growing up, whether you've got polycystic kidney disease or not. You'll have 5 children in a family, and one will think that they were the child who didn't get enough, and the other one will think that they got too much, and everyone has their own story. So people's lived experience actually influences how they think about this condition. Some people think it's not such a bad condition. Other people think it's a really devastating condition, but it all depends on how you've experienced the condition in your family. And how you experience it often influences the way you make decisions about what to do. So there's one thing I just wanted to raise the issue tonight, which is about how people can use genetic information. And some people use genetic information in regards to family planning. I think it's really important to say that if people don't use genetic information for family planning reasons, it doesn't mean that they shouldn't have access to genetic testing. It's really important that people can have access to genetic testing regardless of what their family plans are. So some people want to have genetic testing just to know, and just to know what gene it is that's causing the condition in their family, rather than using it for family planning reasons. And I think in our profession we all talk very lightly about family planning, and we say, oh, you can just have PGD, which is where people can go and have IVF, and they look at the embryos to see whether they have got the gene for PKD, and they only put the embryos back that don't have the PKD gene. And it sounds like a bit of a walk in the park. Well, it's not. It's hard. It's physically and emotionally taxing. People tell you it's really difficult, and it's expensive. So I think, whilst we can all talk about family planning and, PGD, it's actually quite complicated. And also that people have a right to have access to genetic testing, even if they're not going to use the information for family planning reasons. So sorry, I just get on my real hobby horse about that one. The other thing we know about people is that some people are ready to have genetic testing and some people aren't. There's this psychological theory called readiness, and you go through these different stages. Contemplation, pre-contemplation, contemplation, whatever. But, you can see people, sometimes they come into clinic and you can see they're a bit hesitant, and I might say, well, do you not feel ready? And they'll say no. And I say, so how will you know when you're ready? What does that look like when you're ready? What has to happen before you might be ready? Because they'll say they want to have a genetic test, but they're not quite ready to have it yet. So it's really important to help people understand how it's going to look when they know they're ready to have a test. Sounds weird, I know. And the other thing is that families are very funny things. And we don't all get along with our family like we like to think we do. And so often talking about this sort of things in families, particularly inherited diseases, can be very challenging. And we know that from the literature on communication around genetic conditions, that it can actually be really hard for people to talk to their family about it. And there's a whole lot of reasons for that. One can be that they feel guilty, they feel ashamed. Another can be that they don't get on with people, they've fallen out, they don't see people geographical distances. There's a whole lot of reasons. But talking about this sort of stuff can be tricky. So we've talked a lot about genetic testing, and I suppose what I wanted to say is there's advantages and disadvantages, and it's not, you know, everybody thinks about it differently. So the advantages are is it can provide an explanation as to why this is happening in the family, and it can also tell you what gene is in the family, and that can give you information about the course of the disease, and it may also help one day with drug therapy. And it provides information for people. Some people just want to know. And as Andrew talked about before, the mother with the child who actually then said, now this is, I know something I had no control over. It wasn't something I did during pregnancy that caused my child to have this condition. So as parents, the last thing anyone wants to do is to pass something on to their child that might cause a disease or concern. And so we have to acknowledge that in talking to families. It's not just about having genetic testing. And for the individual, there's benefits that it can alter treatment and management. And for families, it can provide, you know, early detection. The thing about predictive testing, just to say to people, is what you can do is once you identify the gene in the family, you can then test the children. Some of them will have inherited the gene and some of them won't. And so the good news for the ones who don't is that they don't have to have any kidney screening or whatever. And the ones who do, they know that they can go into screening, hopefully early detection and sometimes some drug therapy for people. What are the disadvantages? I shouldn't say disadvantages, is maybe not the right word, but what are the challenges? That it does cause distress. We know that. But it's not distress that people don't live with every day. People get upset, but they recover from that. It's bad news, but people learn to live with it, and it's information people want. And as I talked about, we've got family communication problems. The other concern, of course, is that the information will be used in an adverse way, and I'll talk a little about insurance just to finish up. So the thing I wanted to say, it's not either or, it's both. And that's what we can talk about when people come into genetic counselling is, well, this is the good thing about it, and this is, say, some of the challenges about it. Where do you sit? How are you thinking about that? So just to finish up, the other thing that's really important is to contextualise the concerns around insurance, okay? Because I think there's a lot of myths out there about insurance. So private health insurance products are not impacted by genetic test results, okay? There's legislation in place to protect people being discriminated against in regards to private health insurance. But life insurance, companies can use genetic information and will ask for genetic information before people take out policies. Now, I might sound a bit cruel when I say this. So I worked for 20 years in oncology genetics, and so often you'd sit there with women with breast cancer and they'd say, I'm not having a genetic test because I won't get life insurance. And, you know, in your nicest possible way, you'd have to say, look, I'm really sorry, I have to say, but you won't get life insurance because you've actually got breast cancer. It's got nothing to do with the genetic test. It's actually the condition that you've got. So I think there's a lot of myths around insurance. And again, I say this respectfully, but if you've got a condition, probably like polycystic kidney disease, I don't think that you would get life insurance. So genetic testing for someone who has polycystic kidney disease, insurance shouldn't be something that's a barrier for you. So I think insurance companies are using it for people who actually have genetic tests to show they've got a gene before they get a disease. But if you've actually got a condition, you have to disclose it, you have to disclose your family history, and genetic information is one more piece of information that they're using in regards to assessing whether they load a policy or whether they deny a policy. The other thing that's really important to contextualise is only 1/3 of Australians take out private life insurance. Okay, so we're only talking about 1/3 of Australians that this relates to. So most of us would have some sort of life policy on our superannuation, and many people don't take out extra insurances. So this only relates to people who take out extra insurances in the private sector. So I'll finish there. Thanks very much for your time.
So one of the things that we do when we see patients in genetics clinics is spend a lot of time taking their history to talk about their story and also the history in their family, and then thinking very much about what the best test is for that person sitting in front of us. And you know, lots of patients that we see have lots of different conditions because people don't follow the rules, like Andrew said, and sit nicely in one box like the textbooks we read in medical school. And often we think about what tests we can do that can perhaps encompass a number of the things that they have, but also that some conditions are more complex than what our testing can offer. And though we know that things like polycystic kidney disease often have a single gene cause, then there's things like nephrotic syndrome that sometimes have a single gene cause, and then things like endometriosis that perhaps, well, we certainly don't understand the genetics on very well. And so we talk a lot to people about their individual things and which test would fit them. And sometimes I order a test that is for one particular gene or two particular genes for PKD. Sometimes we do testing across the whole genome looking for a number of different potential things. So that's why when we talk about this, that it's really individualised, that all of us have a different set of instructions, different kind of DNA code, and so kind of trying to fit everybody into one box is really tricky. And you know, so when we see patients in our genetics clinics, we take an hour with the patient because it takes time to get their story, to talk about what the options are and talk about what's best for them, like Mary Ann said, to go through the counselling process of what testing they may or may not be interested in having information on at the moment. So it's certainly things we talk about. So you don't need to explain all of that to specialists. You can just turn up to us and we can do some work.
Question for yourself, Andrew, if I may. You mentioned during your talk that we can't begin to treat what we don't understand. Do you think we understand enough about PKD or ADPKD to treat and manage effectively today?
Now that is the question, isn't it? So I think there's a dogma about, you know, the gene conditions being, you know, that were inherited where it was, you know, there's a gene that's got a change in it that stops the gene working, and it's about just one protein that it's the, you know, the blueprint of, and we fix that, that's the end of it. That story has rapidly unraveled. We are very complex systems. Each of every one of our cells is a complex system. And in PKD, about every 12 to 18 months, the intracellular, or the cellular landscape of what all the different processes are that seem to relate to why cysts develop because of these changes in a small number of genes that seem to signal in a similar way, gets bigger and bigger and bigger and bigger. And most recently, the most effective diagram I've seen of the underlying pathogenesis of ADPKD actually is approaching it from a cancer perspective, looking at multiple different pathways. Are we ever going to know enough about ourselves? No. Do we know a hell of a lot more than we knew 30 years ago? You betcha. I think we're on the very cusp of transforming here. I think when they first described the genes that are associated with ADPKD, specifically PKD1 and PKD2, what, 25?
1990.
Oh, yep, 25-ish years ago. Do you think that they thought that at that time, when they identified those genes, thought that, you know, things like calcineurin inhibitors or tolvaptan, these kinds of agents would be the agents that might actually be put into clinical trials? I highly doubt it. There's more information to find out. We're a hell of a lot further along the way than we were, but importantly, I think the work that people like Amali's doing here is going to help us to continue to unravel this because it's a lot more complex than I think we thought it was even 3 years ago.
And if you look at other genetic conditions, so as I said, my background is really in cancer, but you know the genes BRCA1 and 2, you've heard about them. So they were discovered in the late 1970s, it does show how old I am, and from there, I can't tell you now the progress that's been made, and using those models to then come with a condition like polycystic kidney disease. So the genes were found, no one knew what to do. Then it was discovered that if you have a particular gene, you have a particular type of cancer, so there's a biological there's biology underneath it. Now there's drugs developed to give people to prevent it. So if you think about other conditions and models, then people like Andrew and Amali will look at those models and think, okay, well with PKD, what can we do? It'll be much faster, much, much faster. Yeah.
Depends what kind of twins. So, if you're identical twins, then you should have pretty similar genome sequences. If you're twins that they call them fraternal twins, so where there might have been two eggs that were, and then there's two sperm that fertilise them, then they are like siblings essentially, so that sometimes we're quite similar to our siblings and sometimes we have differences in our DNA to our siblings. So it depends.
I've recently seen in clinic a different inherited kidney disease. Same condition, a rare tubular disorder, 3 pairs of identical twins, both with the same, it's a recessive kidney disease. And the important thing is that, exactly as Amali said, they're in all other ways, genetically expected to be, as close to the same as you can get. Their experiences and their symptoms are different. So even the genetics is a significant proportion of determining your risk of having a condition, but it may not be everything in terms of your personal experience of how it exhibits and the symptoms that you have. And I was completely naive to that until out of the blue this happened just fairly recently.
Yeah, so the first step would probably be to go and talk to your GP and say that that's exactly what you want information on, and it's a really great question. And something that certainly in genetic services we're really happy to see. And we see lots of people who are siblings of people with inherited conditions, and then they would make a referral to your local genetic service, and then you would get seen by a genetic counsellor or a geneticist maybe, and then go through the family history. And then it'd depend a bit about your own family and what is known in your family about the condition as to what steps there are next. Sometimes we like to meet maybe someone in the family who has the condition, because generally testing is most straightforward in someone who we know definitely has the disease, because as Marianne explained, the results from the testing are unfortunately not as black and white as Thomas and I would like them to be. And so it can be even more confusing if we don't even know if the person actually has the condition or not. So we usually try to start with someone who generally has the condition in the family, and then we can identify a particular difference and then look in other family members much more straightforwardly in a more black and white fashion. But it depends on the different families that we see. And sometimes there's families where we can't get hold of the person who's the most affected in the family or someone that's affected, and then there's other options that someone would speak to you about based on your particular circumstances. But that's a really, it's a good question, and lots of people who have conditions in their family are really interested to know what their risk is. It's a good question, and it's something that we try to give people as much information about. Does that answer your question?
Yeah, and given that it isn't me with PKD, does that mean that it isn't always likely to be an out-of-pocket expense for me?
Not necessarily. So when we see people in genetics clinics, I guess I can speak for the state, that when we see people, what our policy is that we think about testing, and we would love to be able to afford to pay for testing for everyone, but that's not a decision that apparently I'm allowed to make. And so what we think is this particular test going to change the course of the management for this person that sits in front of me? And whether it changes things because it means that someone can understand the reason that they have a condition, and I think that is a really powerful reason to understand that, there's so much that comes with having a condition, whether it's in your family or whether it's in you as an individual, that understanding the reason is a very powerful reason. Whether it's because someone says, I want to be a donor for someone in my family, but I want to make sure that I don't have it, or I want to make sure that I'm not at risk or my children are at risk. That's a great question, and something that we think is a really good reason to fund the test. Or if someone says, I want to know what my chances are of passing this on to my children. Not necessarily because I might do something to avoid it, but just so that I know what the risk is, then that's a really good question as well. And so there's lots of different reasons. So it's, I guess, less common these days that there isn't a reason why we could fund a test, but it's very much depends on the person that sits in front of us. And that's a different story to what it was probably even 5 years ago. The space has changed a lot.
So working, working with people like Melissa Little. Melissa is a very astute, conservative scientist. I think until about 2 years ago, Melissa was saying it was likely to be about a 50-year time frame, and that's someone who is at the very forefront of this kind of technology. Melissa's on record at the European Society of Paediatric Pathology last year, gave what many had actually argued is the best ever plenary for that society. That she thinks it's now about 25 years. So this is a moving space. I think the first cat off the rank is going to be, rather than a bioengineered kidney where we grow them from your own cells, is going to be a viral implantable kidney, and the phase 1 trials for that are happening at the moment at Vanderbilt University in the US. So I suspect that's likely going to begin to impact our market in terms of clinical trials here in Australia in the next, say, 5 years. But this is moving pretty rapidly. Personally, I invest, it sounds horrible, but literally none of my personal time, research-wise at all, in dialysis. I look after 30 dialysis patients, yes, and I very much care for them, but I do not invest any of my personal time in research activity or anything of that like in dialysis, because I do not think that that is going to be the way forward, and I think that major changes are coming and will change it by the end of my career, much like there was a generation of renal physicians where there was nothing available and then dialysis came. It will end and something else will replace it. I hope that answers it. It is a time for hope, but it's going to take some time coming.
Yeah, so PKD, the genes are definitely big, and that was a big challenge, and there's lots of big genes in the genome that used to be quite challenging to sequence, particularly by Sanger sequencing, which is the traditional sequencing technique, and that's how the whole genome project was done, and that's why it took such a long time. These days, those amazing machines that you saw are very good at sequencing large amounts of DNA, and so the size thing is not so much of an issue, but what was an issue still was that the sequence of the PKD gene, the little code, was really similar to another section of the genome. And so distinguishing the two was really difficult. And even with other similar techniques, so I guess not Sanger sequencing, but other techniques, that was difficult to tell apart. But with the whole genome sequencing, what we think is the difference is that it hits a little bit of a sweet spot, that it doesn't cut the DNA into as small fragments as we used to cut it, so the piece of the puzzle is a little bit bigger, and maybe we get a little corner of the puzzle that's unique, and that can give us a clue. But also that the whole genome sequencing sequences the whole genome, as it says. So we used to just sequence the coding bits of the genome, which is actually only about 2 or 3% of the entire genome. But now we sequence the coding bits, and we sequence the gaps in between those. And we think that the combination of that, getting all of that information and having reads that are bits of DNA that are a bit longer, has meant that the technology, the very clever computers that read the sequences for us, can tell the difference between the PKD genes and the copy genes, the pseudogenes. So yeah, I think that technology has hit kind of a sweet spot there, and it's working well for us. Taken a long time to prove to ourselves, but it certainly seems to be working nicely now. And I guess one of the things that a lot of people say, well, why do you do whole genome sequencing of the entire genome and then you look at 2 or 3 genes? Seems a bit like you're using a sledgehammer to kind of crack a nut. Is part of it is that sequencing trick, and that a lot of the other sequencing platforms have trouble with it, but also that this is a test that we want to be able to afford, be able to offer at a relatively affordable price. And the tricky thing in Australia is that though there's many people that have PKD, there's not the volume that we get in somewhere like America or in the UK. And so if we were trying to offer an affordable test, we'd be collecting samples for a year to get enough patients to fill that machine. But if we can use the whole genome sequencing test that's the same as the heart patients use and the cancer patients and the patients with intellectual disability, we can all lump them together. The lab does the same preparation, and then on the other end we look at different genes, and it makes it a lot more streamlined. So that was another reason why we thought this was a really attractive way to diagnostically test PKD, because it allowed us to streamline some of those processes.
And I think it has been transformative, because as someone who used to request prior to Genome 1, in the Queensland Clinic, we're doing fairly large volume PKD testing. The only other service that we had clear access to was in the United Kingdom, and they would just test PKD 1 and PKD 2. As we keep saying, patients don't conform to nice neat boxes, so actually a significant proportion of the time you were getting negative results, and so then you have to look at another set of different genes that can look like PKD, and so we were cascading, ordering multiple genetic tests, each of which have their own expense, and then there's getting patients back to tell them the results, the clinician's time as well, and so this all adds up. Whereas if you have a single ability to look at all of those genes of relevance to that condition in a single file sweep, one, it increases the efficiency of the test, net, it's actually cheaper, and three, it actually helps patients get answers quicker. So I think from from a clinician user perspective, this is a dream, because before it was a nightmare.
Yeah, so for a DPKD, the one where there's multiple generations of families that are affected, there's two main genes that we know about that are helpfully called PKD1 and PKD2, so we can remember them. And for most people, so probably about 80 to 90% of people who have the picture of PKD, they will have a difference or mistake in one of those genes. There's still about 20% that we don't understand, and some of them fall into those other genes that Andrew was talking about, but then there's a proportion for which we really don't understand, and that's a big part of what the research we do here at Garvan is about. And then for ARPKD, which is the recessive form where parents might unknowingly be carriers and then have a child who's affected, there's one particular gene, and then in the last few years, just because of, I guess, some of the advances of this technology, there's been a few other genes that have been identified as well. But it's a changing space, and so, we'll probably discover that there's other genes that are influencing what we see as well. It's pretty remarkable that you get a little tiny spelling mistake in one gene out of 20,000, and you can get this incredible disease. It's
I personally have a mutation for melanoma.
Yeah.
And it's on P16. And therefore that is other.
Yeah, our bodies are complex.
We're all little clouds of genetic changes.
That's right.
So having genetic changes is actually normal. Marianne, correct me if you think I'm saying something out of turn. But I think, but this is what we often tell our patients as well, is that actually having genetic changes is completely normal. That's what makes us look different, have different blood groups, have different skin, hair, eye colour, all these different things. We all have a number of genetic changes in genes. I think it's estimated somewhere in the range of 40 to 80 that could be deleterious genetic changes that could result in disease, that could harm us. We all have 40 to 60 genetic changes that we didn't inherit from our parents, that are spontaneous and brand new in you, that your children may inherit from you. This is how new genetic change happens and drives evolution, amongst other things. So we've gone from a time where we had no visibility about these little clouds of genetic changes within all of us to complete visibility, and that change is confronting and scary.
And it's happened, in a very short space of time.
So reframing what's normal, I think, is one of the biggest things that we do with our patients. That just because you have this condition or any other condition doesn't mean you're anything less than a normal healthy person. It's just a different part of you.
Well, thank you. Thank you very much to all three of our speakers. I'd just like to actually end and ask one more question to a friend of mine, Ben. So Ben is a patient, and he's had whole genome sequencing for his condition. And I was just wondering, Ben, if you could just share in a few words what it has meant to you and what you got out of it.
So Nicole hasn't put me on the spot here. She has ahead of time talk to me about this. And it's something that I share fairly regularly as well. So I'm a bit of an oversharer. I'm one of those kind of people who tells a little bit too much and also seeks information. So it's a little bit ironic, really, the situation that I'm in. So about 3 to 4 years ago now, I got a diagnosis of Stargardt disease, which is an inherited form of macular degeneration. So what this means is that I will slowly, over the next few decades, progressively lose my central vision. So I'm a little bit unusual, though, in this disease. So this is usually what we call a juvenile onset disease. So by rights, with this condition, I should be blind right now. Now, I'm not. I can actually still legally drive, although I have to get a medical certificate every single year to make sure that I still am allowed to drive. But the irony here is not lost on me in that I used to teach at university on what we call incidental findings. So these are when you look in someone's genome and you find something that, hey, we found something here. So say you're looking at a 20-year-old without renal disease and you find a PKD1 deleterious mutation, they're very likely to get renal disease later on. So in actual fact, I had one there all that time that I was teaching about these things. I also happen to be a scientist who works in our clinical whole genome sequencing facility. I also happen to have a master's degree in genetic counselling. So I'm the scientist, I'm the counsellor and the patient in this situation. So I was lucky enough to actually, well, depending on your perspective, lucky enough to get my whole genome sequenced probably about a year ago now through a research grant on retinal dystrophies within the Garvan here. Now, at my diagnosis, I went and saw a clinical geneticist who happened to be a colleague because it's a very small field. And they offered genetic testing at the time. And I didn't get that done at the time. I didn't see a lot of benefit. I didn't have children at the time. Oh, no, I did actually. I had a 3-year-old at the time. A lot of other things going on. And so ultimately, I wanted to get my whole genome done because I saw the utility in having that done. And this is speaking as a person, as an individual, as a patient, not as a representative of Genome 1 or Garvan. And so I got this done last year. Now, the interesting thing about this is Stargardt disease is a recessive condition. So like autosomal recessive PKD, you need 2 mutations to get the disease. Now, the traditional testing that we've done, in my case, would be to look at the gene ABCA4, which is the only gene known to cause Stargardt disease. So we really were looking at my genome for one gene. We looked at that gene and we found one variant on one of my copies which caused what we call a stop gain. So this means that instead of having the entire protein made, and it's an enzyme, instead of having that entirely made, it stops really quite early. So it stops around about 1/3 of the way through the gene. So I get no functional protein there at all from that copy. Now, that's okay. 1 in 40 to 50 of us, so probably one out of everyone here, will have a mutation in that gene. So it's not an uncommon thing, but you need to have children with another person with a deleterious copy as well. And then there's a 1 in 4 chance. So it's around about 1 in 8,000 people who get this disease. So I really am truly a rare disease patient. Now the problem with me is that we couldn't find the other one. And to this day we still can't find the other one. So I am clinically diagnosed with Stargardt macular degeneration. But we still don't know what the other allele is. Now, the additional irony here is the only test that will tell me what that other mutation is whole genome sequencing. So this is currently so I now have my genome, I have my data, I've gone through ethical release of my genome. I was not involved in the testing at all, as hard as that was. So I had other colleagues doing all of this stuff. And then I've gotten release of this data to me. So now my job really, as a private citizen scientist, is to try and find what that other mutation is. And more than likely, it's one of these things in the junk DNA. So we look at our DNA, so Amali talked about exons and introns. So what happens is our exons are what tell our bodies what to make, and the introns get basically cut out and thrown away. You can get a mutation in one of these introns, which means that it doesn't get cut away properly. And so these aren't always, more than often, these don't 100% change that product. And so you will get some residual leftover gene function. And my belief really here is that I have one of them, which is why I've got late onset of my disease. So I have a mutation that has left some leftover function there, which has rescued my disease to some degree. So now it's just a matter of finding where that is. And for me, the personal drive for this is really mostly curiosity. There's nothing that can be done in my disease right now. But much in the same way that renal disease is a single organ disorder, so is macular degeneration. So the same things that Andrew was talking about with bioengineered kidneys, very much the same research is happening in macular degeneration as well. So I've spoken to experts in this field, and they seem to think that within the next 10 years, there's going to be some sort of curative, curative but potentially not restorative therapies. So, you know, it's not all doom and gloom when it comes to these things that traditionally, as a germline scientist, so someone who looks at inherited diseases, a lot of the argument that people say is, well, who cares whether you know, you can't actually do anything about it. I think that's changing. And I think we're going to see that rapidly change now. So there's curiosity for me. There's also the, as the lady at the back here was talking about with family planning. I already have children. I'm not planning on having any more. God, I hope I don't have any more. But I would like to know whether they're at risk. So the risk to my children right now is 1 in 100. So it's not an insignificant risk there. So I obviously have a mutation. My wife has a 1 in 50 chance of having a mutation as well. So there is a 1 in 2 chance that she would pass on that mutation as well. So 50 times 2 is 1 in 100 risk that my children would have macular degeneration as well. And potentially, if I've passed on a really bad copy and not my not so bad copy that we can't find, and my wife has passed on a bad copy as well, then potentially this could be a juvenile onset Stargardt disease. Now, I'm not particularly concerned about that. I'm one of those people who think that 1 in 100 is actually not a bad risk. You know, 99 out of 100 chance that they don't have that. So it's all about, as Mary-Ann would tell you, risk perception as well, and people perceive risks in different ways. So that's sort of my story, which is a little bit, I think, unusual, but also gives me a fairly nice perspective on the whole thing. So, yeah, thank you.
All right, thank you, Ben, for sharing, and thank you all for coming. Please, can you stay longer, speak to any of our speakers, have some more. There's plenty of food and drinks, and please don't forget to fill in any of those survey forms to see if you found this session useful. And thanks again to PKD Foundation and Garvan. Thank you.
Can I just add our thanks to Nicole for helping to organise it together with Charmaine from PKD Australia, to thank Mary-Ann, Amali, Andrew and Ben for speaking to us, for showing us around, and to thank Genome One and Garvan. I'd also like to make a special mention of Michelle Tarr, Dr. Michelle Tarr. Can you just put your hand up, Michelle? She's the PhD student that I was talking about, and we're really thrilled that she's come back to PKD. She almost got away, but she's back now. So we're really thrilled about that. The other thing, Andrew was talking about the KidGen conference in Sydney. Unfortunately, we as mere community members are not able to go to it, but we have got a way of being there, 3 p.m. to 5 p.m. on Sunday, 9th of September at the International Conference and Convention Centre here in Sydney. We will be having another one of our patient seminars, so there'll be talks about treatment, about diet, about new research, so please come along. If you can remember that website there, you're better than I am, but you can please sign up. You can find it through our normal www.pkdaustralia.org. Please register for it, and I hope we see you there. It'll be very interesting, as they always are. So please, thank you very much, and please join with me in thanking our speakers.